today's topic is viral gastroenteritis. Viral gastroenteritis, mostly children and infants are affected by these viral agents. As a result, those affected infants and children are suffered from diarrhea, vomiting, and in severe cases, we can see dehydrations. In gastroenteritis, there will be inflammation of the lining of gastrointestinal tract, which includes stomach, large intestine, and small intestine. To cause gastroenteritis, not only viruses, bacteria, fungi, and various other parasites are also responsible to cause gastroenteritis. When there is an involvement of viral agents to cause gastroenteritis, we call it as viral gastroenteritis. The main sign and symptoms of viral gastroenteritis is diarrhea, vomiting, which is associated with dehydration. By definition, gastroenteritis means inflammation of the lining of gastrointestinal tract, which includes stomach, small intestine, and large. When there is an involvement of viral agent which are responsible to cause gastroenteritis, we call it as viral gastroenteritis. In common word or in layman term, we call viral gastroenteritis as a stomach flu, in which our stomach will upset. Some of the viruses, they are responsible to cause viral gastroenteritis. And about 3/4 of all the infective diarrhea is caused by viral agents. Viral gastroenteritis, it is second most common viral illness, which comes after upper respiratory tract infection. In developing countries like Nepal and India, this viral gastroenteritis is one of the main cause of death of infants who are malnourished. So this viral gastroenteritis is one of the serious problem of developing countries, like most of the South Asian region. Rotavirus, this is one of the potential viral agent which is responsible to cause viral gastroenteritis. This rotavirus is responsible for 3 to 5 billion diarrheal episodes in children and infants annually worldwide, which results in about 1 million deaths, mainly in Asian and African subcontinent. Besides rotaviruses, several other viruses are found in the gut, but only few are associated with gastroenteritis. So these are the name of the viruses which are responsible to cause gastroenteritis. The most potent viral agent which is responsible to cause gastroenteritis is rotavirus. Then comes adenovirus type 40 and 41, caliciviruses, Norwalk-like viruses, astroviruses, and finally coronaviruses. All these viruses are responsible to cause viral gastroenteritis, but the most potent one is rotaviruses and adenoviruses type 40 and 41. While these caliciviruses, Norwalk, astro, and coronaviruses, their frequency is very less to cause viral gastroenteritis. These are some of the pictures of rotavirus, Norwalk virus, adenovirus, and astroviruses. Besides these viruses, these are the name of the viruses which are responsible to cause viral gastroenteritis. Besides these viruses, in our gastrointestinal tract, there is presence of some other viruses which are not responsible to cause viral gastroenteritis. Can you name those viruses which do not cause gastroenteritis, but they are present in our GI tract? Kamala Adhikari? Kamala?
Yes, sir.
Can you give examples of the viruses which are present in our gastrointestinal tract, but they do not cause gastroenteritis? Influenza. Okay. They found in primarily it is the respiratory pathogens, but sometimes they can go to our GI tract also. What are other viruses? Yamuna Disha Magar?
Yes, sir.
Can you give name of one virus which can found in our gastrointestinal tract, but it is not responsible to cause gastroenteritis?
Sorry, sir. No, sir.
Poliovirus is present in our gastrointestinal. Okay, there are other viruses also like Coxsackie virus, which is present, can be found in our gastrointestinal tract, and these viruses are not responsible to cause viral gastroenteritis. But if you have to remind the name of these viruses which are responsible to cause viral gastroenteritis, there's a mnemonics RNA square C square. RNAC, R for rotavirus, A for adenovirus, another A for astroviruses, then RN, Norwalk-like viruses, and C2 for caliciviruses and coronavirus.
के भाइरस भन्छ त्यो ज्याइटिङ हुँदैन डायरिया नकज गर्ने
poliovirus, Coxsackie virus. Now, today we are going to discuss only about rotavirus. This rotavirus, it belongs to family Rheoviridae. Rotaviruses, they belongs to the family Rheoviridae. These rotaviruses are RNA viruses, and the viral RNA genome, it is double-stranded RNA. These rotaviruses, they are named or called as rotavirus by scientists because when we observe the morphology or structure of this rotavirus under electron microscope, we can see a wheel-like structure. On the surfaces of these viruses, there is presence of viral proteins. These viral proteins, it appears as a spikes which are penetrated in the hub of a wheel-like structures. Thus, these rotaviruses are called as rotavirus since their morphology is similar to like that of wheel. This is the structure of rotavirus. These viruses are, their size is very small, which enters 60 to 80 nanometers. These are RNA viruses, known is double-stranded RNA. They are all together 11 number of RNA segments are there in case of rotaviruses. These are the 11 segmented double-stranded RNA genome of rotaviruses. Its viral genome, that is double-stranded RNA, that coded by viral protein. Segment number 1 is coded by viral protein 1, 2 is coded by viral protein 2. Segment number 3 is coded by viral protein, and so on. These viruses, they are non-enveloped, and the viral RNA genome, it is surrounded by triple layered icosahedral protein. The viral RNA genome is coated by 3 layers of proteins, icosahedral shaped protein. This is the innermost layer. The innermost layer is made up of viral protein 1, viral protein 2, and viral protein 3. Then middle layer, it is composed of viral protein 6. This bluish circular structure is the middle layer, which is made up of viral protein 6. And the outermost layer is made up of viral protein 7 and viral protein 4. Viral protein 7 and viral protein 4. These are the 3 distinct layers which surrounds or which protects the viral RNA genome. They are all together 6 viral proteins are present. Those viral proteins are named or numbered as viral protein 1, viral protein 2, viral protein 3, 4, viral protein 6, and viral protein 7. Then there is presence of non-structural proteins. These non-structural proteins are named as non-structural protein 1 to non-structural protein.
Excuse me, sir, what's the difference between the two?
You mean difference between viral proteins and non-structural proteins?
Yes, sir.
Viral proteins are important, and these proteins are, this protein, it helps the virus, that is rotavirus, for its infectivity. For example, this viral protein 4, it is a spike-like projections which helps the virus for attachment to the host cell receptor, which is present in the host cell. So these viral proteins, it helps the virus to maintain its integrity, while non-structural proteins, it has got other functions. This picture, this is the electrophoresis of the RNA segment or RNA genome of this rotavirus. There are 6 viral proteins and 6 non-structural proteins. This electrophoresis pictures what I'm trying to show is there are altogether 11 segments of RNA genome of this rotavirus, and each RNA segment of this rotavirus, it is coded by a specific viral proteins. Here you can see this is the first RNA segment of RNA virus, which is coded by viral protein 1. Then this RNA segment of 2, 3, and 4, they are respectively coded by viral proteins 2, 3, and 4. Next comes RNA segment number 5, which is coded by non-structural proteins. Then we have RNA segment 6, which is coded by viral protein 6. Segment 7 is coded by non-structural protein 3, and RNA segment 8 is coded by non-structural protein 2. Segment number 9 is coded by BP7, while RNA segment 10 is coded by non-structural protein 4. RNA segment 11 is coded by non-structural protein 5 and 6. Rotaviruses, they can be classified on the basis of antigenic compositions and binary system of typing. The first classification method of these rotaviruses, it is based on antigenic compositions of the group specific viral protein 6. On the basis of this group specific viral protein 6, these rotaviruses, they can be classified into 7 major groups. That is rotaviruses A, B, C, D, E, F, and G. Among these 7 different groups of rotaviruses A, B, and C are responsible to cause viral gastroenteritis. Those rotaviruses which are responsible to cause viral gastroenteritis are rotavirus A, B, and C type. Rotaviruses A and C, it is responsible to cause viral gastroenteritis in infants and children, while rotaviruses type B is responsible to cause gastroenteritis in adults. According to the viral protein 6, rotaviruses can be divided into 7 major groups, rotavirus A to rotavirus type G. Among these 7 different types of rotaviruses, only 3 are associated to cause viral gastroenteritis. Among 3 different types of rotaviruses, A, B, C, they are the potent agents which are responsible to cause viral gastroenteritis. But rotavirus type A is the most common and potent viral agents which is responsible to cause viral gastroenteritis in neonates and children. Rotavirus type A and C is responsible to cause gastroenteritis in children and infants, while type B causes gastroenteritis in adults. Another classification method of rotavirus is binary system of typing, which is based on viral protein 7 and viral protein 4. These are the typing methods. 2 typing methods which are used to classify rotaviruses. One is based on the viral protein 6, and another system which is known as binary system, which is based on the presence of viral protein 7 and viral protein 4. The mode of transmission of rotaviruses is mainly by fecal-oral route. Its prevalence is common during winter seasons. When a person come in contact with the contaminated hands, surfaces, and other objects, these rotaviruses, they gain accesses through our body and cause viral gastroenteritis. The infective dose of rotaviruses, it varies from 10 to 100 viral particles. The feces of an infected person, it may contain about 10 trillions of viral particles per gram of stool sample of the infected persons. So when a person comes in contact with these rotaviral agents, or when a person take a food which is possibly contaminated by these rotaviruses, these viruses gain accesses and they reside in our gastrointestinal tract, mainly small intestine. Our small intestine will be affected by these rotaviruses. The case is very rare. Some of the rotaviruses can also be transmitted to human beings by respiratory route also. When a person vomits, the vomit particles, they are exposed in the surrounding air. And when we inhale the air droplets, which contains the viral particles of rotaviruses, these virus agents gain access to our gastrointestinal. So mode of transmission of these rotaviruses through respiratory route is not common, but the most common route of transmission of rotaviruses is through fecal-oral. Viral gastroenteritis, which is caused by rotaviruses, it is one of the serious cases in developing countries, which results maximum number of deaths of infants and children in developing countries, like most of the part of Asian countries and African countries. These rotaviruses causes viral gastroenteritis in Asian and African countries, where it results death of many children. Now, the pathogenesis of rotaviruses, its mode of transmission is through fecal-oral route, and they resides in the small intestine. These viruses, they resides in small intestine where they take nutrients which is present in our small intestine, and these viruses, they multiply at the tip of the intestinal villi of small intestine. After replications, these rotaviruses, they produces their viral progenies within 10 to 12 hours. After producing their viral progenies, these rotaviruses, they are released into the lumen of the small intestine, where these daughter viral progeny, they invade other neighboring cells of small intestine. This is the biopsy picture of small intestine, where you can see the atrophied villi of at the tip of the small intestine. And here we can see these infiltrations of mononuclear leukocytes. At the tip of the villi of small intestine, these viruses, they damage the tip of the villi of small intestine. As a result, it causes damage to the cell, which leads to malabsorptions of various type of nutrients and electrolytes which are required for our body, along with water. As a result, a person will be suffered from diarrhea along with vomiting. If diarrhea and vomiting is not controlled and the case becomes serious, it may lead to dehydration also. The main signs and symptoms of this rotaviral gastroenteritis is mostly diarrhea, vomiting, and these rotaviruses, they have got a very short incubation period, that is 1 to 3 days. After 1 to 3 days of incubation period, these viruses, the person who is affected by these rotaviruses, we can see vomiting and profuse watery diarrhea. The person who is affected by these rotaviruses, we can see a dehydration, which is very much serious than bacterial infection. We can see the infected persons have got a mild fever. Rotaviruses, it is responsible to cause gastroenteritis in all the age group, but the most severe symptoms can be seen in neonates and young children, which is caused by rotavirus type A only. 6 types of rotaviruses are there, but the most potent is rotaviruses A, B, and C, and only rotavirus type A and C is responsible to cause viral gastroenteritis in neonates and young children. So here I'm talking about rotavirus type A and C. When adults are infected by these rotaviruses, it causes or it produces asymptomatic infections. In adults and older children, it causes asymptomatic type of infections. In persons who are immunocompromised or whose immune system is debilitated, in such individuals, we can see a symptomatic type of viral gastroenteritis. If a person is infected by these rotaviruses time after time, in such conditions, viral gastroenteritis may be asymptomatic one. Mostly, children are affected or infected by these rotaviruses. And we can see a presentation of diarrhea, vomiting, along with fever. And in severe cases, we can see dehydration. The main site of replication of these rotaviruses is our gastrointestinal tract, particularly small intestine. The tip of the villi of small intestine is the main habitat of these rotaviruses. Besides gastrointestinal tract, these rotaviruses can disseminate to various other parts of our body, like liver and central nervous system through blood. So if viral gastroenteritis, it remains for 5 or more than 5 days, it produces a purging, watery diarrhea, and it may lead to dehydration. Now, laboratory diagnosis. Laboratory diagnosis of rotavirus. It helps to identify or detect the causative viral agent of viral gastroenteritis, in which we will measure or quantify the concentration of viral antigen and viral agents. By using ELISA technique, we can also detect the viral RNA genome and see the presence of viral proteins along with viral RNA also. Here, we have to collect the specimens. The best specimen in case of viral gastroenteritis, which is caused by rotaviruses, is feces or stool specimen. The causative agent, that is rotaviruses, that can be detected in the stool specimen of the infected person. Stool is considered as the ideal specimen for laboratory diagnosis of rota. If we have to observe the morphology of virus, we can use electron microscope or cryo-electron microscope can also be used to see the detailed internal structures. By using this electron microscope, we can see the viral RNA genome along with the viral glycoproteins, that is structural and non-structural viral glycoproteins can be seen. Rotaviruses, it is very difficult to grow these rotaviruses under laboratory conditions. If we have to, but for research purposes, we can grow these rotaviruses under laboratory conditions. Among different type of rotaviruses, only rotavirus type A can be cultured in laboratories. For that, we have to use the cell lines of green African monkey kidney cells. Green African monkey kidney cells is the cell lines in which rotaviruses can be cultured. To culture these rotaviruses in these green African monkey kidney cells, we have to add a little amount of trypsin. This trypsin will lyse the viral proteins, viral glycoproteins, or viral, we can say viral envelope, and it promotes the growth of these rotaviruses. In the stool specimen of the infected persons, viral antigens can also be detected by ELISA or latex agglutination. RT-PCR can be used to observe the viral RNA genome. Rapid test can be used in which detection of viral antibody against these rotaviruses, that is IgM and IgA antibodies, can be detected. The rising titrations of IgM and IgA antibodies in the infected person's serum, it indicates that there is the infectivity, our immune response is higher in such infected individuals. Typing of these rotaviruses can also be done on the basis of, on the basis of G and P phenotyping. It is mainly based on the RNA sequencing of these segmented double-stranded RNA of these rotaviruses and neutralization test. Generally, for treatment of the cases of these treatment of gastroenteritis, which is caused by rotaviruses, there is not any specific treatment is there. Viral gastroenteritis caused by rotaviruses, it resolves over time. After 7 days of infections by these rotaviruses, the viral gastroenteritis will be cured without any specific treatment. These are viral agents, so antibiotics are not effective against viral gastroenteritis. We can partly decrease the cases of viral gastroenteritis, which is caused by rotaviruses. It can be little bit diminished, or we can control the cases of viral gastroenteritis by giving probiotic therapy. The main goal of treatment of viral gastroenteritis, it is to reduce the symptoms like diarrhea and vomiting, so that a person will not be affected by complications like dehydrations. If dehydration is not reduced in case of viral gastroenteritis, it may lead to death of infected. Dehydration, severe dehydrations in case of viral gastroenteritis, it can be treated by giving ORS. If there is severe dehydrations, we can give intravenous fluid also. For preventions, there's not any specific factors are there. The first thing is we have to improve our hygienic conditions. One should wash their hands properly, and while they cook food, those vegetables and fruits, it must be washed properly and cooked properly before consuming such foods. In community level, proper health educations and health awareness against these rotaviruses must be given to the general people, and consumptions of clean drinking water is important to prevent ourselves and our community from these. The specific factors for prevention from rotaviruses is breastfeeding. In the mother's milk, there is presence of secretory immunoglobulins, that is immunoglobulin A. This immunoglobulin A, it helps to provide passive immunity against rotaviruses in case of newborns and children. In mother's milk, there is presence of complex carbohydrate, that is lactadherin. This lactadherin is present in the mother's milk along with IgA immunoglobulin. It goes to the gastrointestinal tract of infants or newborns, and that attaches to the small intestine. The rotaviruses, it attaches, mainly these rotaviruses, they attaches to the intestinal villi of small intestine. This lactadherin, a complex carbohydrate molecule which is present in the mother's milk, it mimics to the intestinal villi of small intestine. As a result, these rotaviruses, they think that lactadherin is the villi of small intestine. Thus, these viruses, they attaches to the lactadherin, and they try to feed on those carbohydrate molecules, that is lactadherin. And these viruses, they can't deteriorate the villi of the small intestine. And these lactadherin particles, they are expelled out along with the feces when a person defecates, when the neonates or newborn babies defecates. That's why, as a result, the virus particles of rotaviruses are excreted through feces, and partly this lactadherin helps to protect newborn babies, which is present in mother's milk. So proper hand washing and sanitization practices is the most important thing which prevents us from infection by this rota. So while washing hand, one should use soap and warm water, and one have to wash their hand properly for at least 20 seconds. Rotaviral vaccines is present. The first generation rotaviral vaccines was developed in the year 1999. These rotaviral vaccines against rotaviruses are not effective. The first generation rotaviral vaccines are not effective because that rotaviral vaccines, is responsible to cause intussusception, and it is allergic in nature. That's why later on, a second generation rotaviral vaccines was developed by scientists in the year 2006 and 2008. Those rotaviral vaccines are Rotatec, and these vaccines are safe, comparatively safe than the first generation rotaviral vaccines. Rotatec vaccine is given in 3 doses at time interval of 2, 4, while Rotarix vaccine, it is given at 2 doses and time interval is 2 and 4 months. These rotaviral vaccines are live attenuated vaccines which are administered orally, and these vaccines provides heterotypic immunity against these rotaviruses. Now, some few questions are there. What is VGE? Prarthana Kimire?
Viral gastroenteritis, sir.
Yes. What is VGE?
Inflammation of lining of GIT as a stomach flu.
Mainly which part?
Upper
Enteritis is the inflammation of stomach,
small
intestine.
intestine, large intestine.
In case of infection by these rotaviruses, stomach and large intestine, they are spared, while only small intestine will be affected. Right, Prarthana? Only small intestine will be affected.
Only? Okay, sir.
Small intestine. Small intestine को नि कुन part हो? जुन intestine मा पाइने भिलाइ छ नि, त्यसको जुन tips हुन्छ, that will be affected. Besides rotavirus, which other viruses are responsible? Raksha, besides rotavirus, which other viruses are responsible?
Adenovirus,
Yes,
calicivirus,
yes.
Norwalk,
Norwalk,
astrovirus, and coronavirus.
Coronavirus. What are the common symptoms of viral gastroenteritis? Sakar Deep.
Yes, sir.
What are the common symptoms of viral gastroenteritis?
Diarrhea, vomiting, fever,
Okay. Then
dehydration.
dehydration. Quench of thirst. These are the signs of dehydration. Excessive thirst, infrequent urination. Urine will be dark colored, dry skin. A person feel lethargy, dizziness, and if there is severe dehydrations, it may lead to faintness of the affected persons. Who are the susceptible person from viral?
Dehydration.
Who are
Viral infection.
the susceptible persons?
In developing countries, mainly neonates, infants, and older ones.
Mostly those whose immunity is compromised or debilitated. These persons are susceptible to infection by these viral agents which are responsible to cause gastroenteritis. Mainly neonates, children, and persons whose age is above. That's all. Classification, structure, pathogenesis, and laboratory diagnosis of rotavirus. Thank you.